Swayam Prabha. Digital India. Educated India.
Welcome. We are into our lectures on neural encoding and decoding, and in the previous lectures we talked about statistics of spike trains, the variability in spike trains, that spike trains generally follow a Poisson process like characteristics, and the spike counts follow generally a Poisson distribution over a certain interval. And we also discussed about defining stimuli as random variables or random processes depending on the situation, and how we will be treating the stimulus world in the case of encoding and decoding as a random variable or random process, and how we will then try to connect it with another random variable or random process, which is the spike trains. When I say random variable, the response space, if it is simply spike count over a certain window in response to a stimulus whose parameters are changing, then the response is simply a random variable, which is the spike count in response given a particular stimulus. However, in general it will be a random process because we are recording from spike trains, and we discussed the issue where the response measure is based on a response rate in a time window, and the size of the window can vary and go down to very small values, and even be a continuous time process or a point process in continuous time. So, in order to go on further, what is it that we want to understand about the relationship between, let's say, the stimulus and the response? One way to approach is a descriptive kind of relationship, which simply tells us what the receptive field of a neuron is. So, if we are recording from a sensory neuron in an intact animal, let us say either an auditory neuron in the auditory pathway, which you are familiar with in terms of an hierarchical pathway, or the visual pathway, or somatosensory. These three, as you may recall, are the parameterizable or parametric stimulus, physical stimuli, and we also have the chemical stimuli, which are olfaction and gustation, where we do not have a parameterization of the stimulus space. So, these have no parameterization possible so far. So, essentially in these cases we have to resort to this idea that a neuron responds to odorant A and odorant B of a set of odorants A, B, C, D, let us say, and maybe we can also add to it that to the odorant A, the neuron responds with a certain number of spikes per second, and to the odorant B with another certain number of spikes per second, or even talk about a temporal pattern of spikes, that is by changing the time window of binning of the response space. So, along those lines, even in the auditory and visual and somatosensory stimuli, we can have a descriptive definition of the receptive field. That is how it actually all started. So, for example, we can have let us say we are recording from a visual neuron, and we are representing in this rectangle the entire visual field of view in front of us, and or the animal from which recording is being performed, and we are essentially getting spikes, and now we start to flash lights at a small resolution along this entire window, and we measure the number of spikes occurring to light flashes in each of those positions x and y. And we find that in this particular region, that is when we have flashes of light here or anywhere within this circular region, the neuron responds to the light flashes. Then this circular or region is sort of the receptive field of the neuron that we are recording from. So, there are a few things here. One is the number of spikes. We said that the neuron responds, and so with it, as in we discussed in the previous lecture, we must have a response measure. That is essentially, for now, it is the time window or time resolution of the response. And the other thing is how to understand that there is a response. So, let us think of it in this way, that there are spikes occurring in neurons spontaneously as well. So, that is without any stimulation, due to simply noise or other fluctuations going on from other connections coming in where there is activity, we have spiking as a baseline. So, let us say we are recording and we obtain spikes at certain windows. So, this time period, let us say, is the baseline, or in other words, during this period we are not doing any stimulation, and let us say in this period the light flashes are coming on, and here also we find spikes occurring at certain intervals, and then it responds again with its spontaneous activity. So, to conclude that there is a response here in response to the stimulus in this particular window. So, this is the stimulus. We have to compare it with the baseline where there is no stimulus, and assume that preceding stimuli even before the baseline are in no way affecting the responses in the baseline. That is, it is indeed a true baseline, I mean theoretically a true baseline, as if there is no dependence on anything happening in the past. So, to compare, since we know that these are random variables, that is the spike count in an equal sized window in the baseline as the stimulus window. So, let us say this is the stimulus window, and this is our baseline window, the cyan and the red, they are of equal sizes. So, we need to compare whether the number of spikes occurring in that same window, in the red window and the cyan window, are different or not. Since these are random variables, we have to take help of statistics, and we also have to do repetitions to get a better estimate, or at least a good estimate of how many spikes we can expect from the stimulus and how many spikes we can expect due to in the baseline period of an equal amount of time. And as I have drawn in this picture, there are 4 spikes and 4 spikes in both cases. So, by just looking at this, we cannot conclude whether there is a response, or even we cannot conclude that there is not a response, that definitely there is no response, that also we cannot conclude. So, it also means that here we are considering the response measure to be the number of spikes in whatever window size we are considering. It may be that by changing the response measure, there may be a way by which we can say that they are different in the baseline and the stimulus. So, that could also mean that there is a response, at least in terms of the spiking happening that we are observing. So, for example, when we are talking of, so let us go to a new page and let us say that the response measure is spike count, or simply the average firing rate, which is the number of spikes by the time window that we have. So, we perform this stimulus presentation over and over again on multiple trials. Let us say we repeat 1, 2, this is the ith trial, let us say we do capital N trials. So, how many trials we need to do also depends on how variable the responses are, and based on knowledge from previous experiments and so on, we have an idea for the structure that we are recording from, how many repetitions are good enough. In other ways, you can also think of it in this way, that if we see that by increasing the number of repetitions, we are not changing the response spike count, average response spike count or average response rate, then whenever at whatever capital N we are seeing a sort of convergence of the rate of spikes, we can consider that we have sufficient number of repetitions. And similarly, preceding each of those repetitions, we will again have a baseline period, which we had in the cyan lines. And so, we have a rate B1 in baseline, rate or spike count B2, I am using it interchangeably here, and rate BI and BN, B capital N. Similarly, we have in the response window R1, R2, Ri and R capital N. So, now we have to ask this question, whether this set of responses or baseline values and these set of responses, they are significantly different or not. So, remember that a response can be an increase from baseline, can also be a decrease from baseline. That is, the neuron may be inhibited due to a stimulus, and it may be inhibited significantly such that the number of spikes occurring is lower than baseline, substantially lower than baseline. Or if a stimulus is exciting it, it may be above the baseline. So, these we do based on a statistical test, hypothesis test, where we simply ask the question whether the mean of the observations R1 to Rn is higher than, or first is different from the baseline observations B1 to B capital N. And we basically now compare the means of these two. So, we have statistical methods for it. Usually, we use a t-test, assuming that the distribution is Gaussian like, which is the case when we have a sufficiently large spike rate, and we assume that the rate is a continuous random variable, then the Poisson converges towards a Gaussian distribution. It can be approximated close to a Gaussian distribution, and so it becomes a test of whether the two Gaussians are significantly different or not. and that depends on how variable the two distributions are. So, let us say this is the distribution of responses where this axis is rate, and let us say this is the distribution of baseline windows where these B values are indicated by the cyan distribution. So, B1 to B capital N are representing this cyan Gaussian window or a Gaussian distribution, and R1 to R capital N represent this red Gaussian distribution. And now we essentially ask the question whether this mean value and this mean value are significantly different or not, and that depends on how much overlapping region we have in this region. That is because that is the region where if we observe a rate in this particular range, then we cannot be sure whether it came from the baseline or whether it is same as the baseline, that is came from the baseline distribution, or it came from the red distribution or the response distribution. Obviously, if the rates are in this range, which it is highly unlikely that the number of spikes, the number of spikes observed come from the baseline distribution, or that there is probably a response in there. And if the observations are in this range, we can obviously be sure that it is coming from the baseline. But again, there may be another window where we can say that the responses, if it were much lower than that, if there was a red distribution on the left, then it would be lower than the baseline distribution. In this case, it is on the right hand side, so we are not worried about that part. So, it is an excitatory response. So, the details of the measures that are involved in calculating this, whether there is a significant response or not, that is a statistical problem, and we have functions in available to us by which we can do this statistical comparison and say with certain confidence or with a certain probability that indeed there is a response, and usually we use a confidence level of 95%, sometimes 99%, and there are variety of corrections that need to be done in terms of how many comparisons are made and so on, which are a separate issue. We will touch on them later on when we are discussing about discrimination. And so, from those kind of tests, we can conclude that there is a response or not. And so, if there is no response, that is, if the rates, the mean rates are the same, or the baseline distributions and the red distribution are highly overlapping, and so we cannot say with confidence that there is a response, then we can only conclude to this point that there is no response if we consider rate as the response measure, that is the overall rate over the entire time window, which is essentially the spike count also during that entire time window of the stimulus. We are assuming that the time windows are constant, are same in the baseline and in the stimulus period and in every trial. So, it may be, as I was saying, that there is a difference in the responses at a finer resolution of time, and that comparison requires further kind of measures, where we will be talking about them later on when we discuss discrimination along the same line, and say that okay, these are discriminated by the neuron in terms of the responses produced and the timing of the spikes. So, for the most part, when we are considering rate response, we do not pay attention to the specific time of the spiking. and so it is, the response is independent of where the spiking occurred, which is what it is when we have a fixed Poisson process, that is with a fixed rate, a homogeneous Poisson process. And in the other case, we can have an inhomogeneous Poisson process. So in the homogeneous Poisson process, we have a fixed lambda in the response period and a fixed lambda in the baseline period, which is what we were comparing here. This is the lambda r estimated from the data, this is the lambda baseline estimated from the data. However, the response may be actually lambda RT, which is an inhomogeneous Poisson process, or as we had also said, that by discretizing time as delta I in the ith window post the stimulus. Similarly, the lambda b can also be lambda baseline as a function of time, or in the delta jth window in the baseline period. And so this requires comparison over the entire time, and those comparisons are different. So the point that we are trying to make is that saying that there is no response based on the spike rate doesn't necessarily mean that there is no response for any kind of response measure. So that needs to be kept in mind. Having said that, we will continue here with the idea of simply spike count or average response rate over the entire time window for the ensuing discussion of receptive fields. So we came to this point of whether there is a response or not based on the response observed or the spike trends observed in terms of flashes of light in that circular region. So let us just recap that. So we have this rectangular region which is representing the entire field of view in front of us, and we have this particular region when there is light presentation in that circular region, then the neuron that we are recording from responds significantly over baseline based on our previous discussion of how to compare, and we can then say that this circular region is the receptive field of the neuron. And this is, although we did all these statistics and measure of response and so on, this is essentially a descriptive kind of receptive field. In other words, we haven't understood the relationship between the stimulus and response. So we have only identified a region in the stimulus space to which the neuron is sensitive. That is a fine thing in the sense that, okay, we are describing the receptive field in some way, but we are not quite quantitatively getting into the idea of the relationship between the stimulus and response. So this brings us the idea of how to model the receptive fields. And before we go into the modeling of the receptive fields, we then also need to understand that the receptive field, the descriptive nature of receptive field that we say by this region in the field of view to which a neuron is sensitive, it is only a boundary. So there may be specific features in the stimulus, that is the way the flashes are occurring overall together in that entire receptive field, to which the neuron may be extra sensitive. So we may be able to parameterize the stimulus in a particular way within that circular region, and then conclude that a particular set of parameters is what the neuron responds best. So for example, since we are discussing about the visual system, in the visual system, what long ago Hubel and Wiesel showed that in the visual cortex, there are neurons that respond to oriented bars, bars of light. So, essentially, if in this region we have a bar of light, let us say we found that round circular receptive field region. Now, we are qualifying it further as to what within that region describes the response of the neuron best, or at least what kind of stimulus within that region makes that neuron fire the most or produce the maximum firing rate, or how varying the parameters within the receptive field, that circular receptive field, the neurons responses are changing. All these are indirectly ways of representing receptive field of a neuron. So, it may be that we have a dark region surrounding a light bar like so, and it may be moving in a particular direction, generally a moving bar, and let us say what Hubel and Wiesel did was that we have this axis, horizontal axis, and this angle of orientation of the bar is theta, and we are measuring responses as a function of theta. So, we have this axis as theta, and if the orientation can vary between 0 to pi, or actually 0 to 2 pi if we consider the direction of motion as well. Now, as we vary theta, we measure the responses of the neuron for each of those thetas that we test. Maybe that 0, then 22.5, then pi by 4, then 75 degree, 67.5 degrees, and pi by 2, and so on. So, basically we are dividing the 0 to pi into 8 specific or 9 specific stimuli. The parameters, that is the theta in this case, is being changed from 0 to pi in discrete steps, and we measured the rate of response for the 0 angle. So, when the bar in that circle is horizontal, and then it is tilted, and in this case it is vertical, and in this case it is again horizontal, and we plot the number of spikes or rate, assuming the duration is same, along at these points. And what we find in these visual cortex neurons that are simple cells, that they respond with a peak at a particular angle of orientation, and this is called this is known as orientation tuning. That is, neurons are tuned to a particular orientation of the light bar, and this also is a description of a receptive field of that neuron. That is, which essentially says that it responds strongest to a 67.5 degree oriented bar with spike count as the response measure. However, this does not answer the question that if there were 2 bars in there, that is, one a horizontal bar. So, let us draw it separately. Let us say this is the round region. Let us say now the stimulus is something that is a horizontal bar and a bar at 45 degrees, let us say, these two together. So, this also can be a stimulus, but the answer to the question that what would the response of the neuron be to this kind of a stimulus cannot be obtained from this description of the receptive field. So, we need better models in order to be able to describe or better definitions of receptive field in order to be able to describe the relation between the stimulus and response in a more complete sense. I am not saying that we will be able to always be able to do this accurately for every neuron, but the objective here is to define the receptive field as a model of the neuron in terms of its response as a function of stimulus parameters or multiple stimulus parameters. So, the first thing about it is that it is not that the response is going to be what the response is at 45 degree and what the response is at 0 degree, and you add them up together. That is not how neurons generally behave. However, if it were to behave in this manner, it would be a very linear kind of neuron, but in general this is not true, and so we require better receptive field models, and that brings in the idea of having models for the neuron to describe its receptive field or its relationship, how it relates the stimulus to its response, to be able to predict that response given new stimuli. That sort of question is what we would be after in terms of understanding encoding and decoding by a neuron. So, in the next lecture we will start with this basic idea of building models. We will start with what I just described as the linear